Throughout your two-part semester, you've learned that a property of cell membranes is regulation. That regulation can be with respect to the flow of ions between the interior of the cell and the outside environment. Ions like sodium ions, chloride ions, magnesium ions, these are charged. And as a result, they can also regulate the flow of electric current through the membranes, as well as electrical potentials across them. Now, all cells maintain electrical potentials across their plasma membrane. This is a topic discussed with respect to membrane potential and the sodium potassium pump in Bio 100. But the nerve cell has a special mechanism for using this potential in order to transmit signals over long distances. And in this chapter, we'll discuss the behavior of neurons and their ability to achieve effective communication.
Here is an example of an organism that utilizes the behavior of the nervous system as a means of immobilizing and acquiring its prey. This is a tropical cone snail. It's a carnivorous organism. Different species will kind of acquire different nutrients, meaning that some will eat worms, some will eat fish, and they hang out within tropical waters. They can also be found in the deep seas off the coast of Florida. And the mechanism or the mode of action in which they can acquire their prey is through the release of toxic molecules. And these toxic molecules can disrupt neuronal control in their prey. So in a fish, which is an example that your book uses later on. And upon injection, the body of that organism will respond with paralysis and death. And something that's really interesting is the pathophysiology of the genus. So meaning, all the different types of cone snails, not just the Geographis that your book describes. It's possible that the cone snails cause death through respiratory toxicity. So imagine if we are messing up the neuromuscular pathway within respiration, that we depend on intercostal muscles and the diaphragm in order to achieve that. So breathing can stop based on any type of, messing up of that particular pathway. There can be cardiovascular toxicity. So we know that there is autorhythmicity when it comes to the heart, but we also know the idea of communication in terms of being able to facilitate those muscle contractions. So we know that both of these things are really important for remaining viable, right? For remaining alive. So there can be respiratory toxicity, cardiovascular toxicity, or even both. And the outcome is based upon the types of peptides in the venom of the snail and the specific type of neuromuscular pathway that they interfere with. So this is a really nice way to think about respiration, cardiovascular function, and cell communication with respect to pathways and what you learned about way in the beginning of the semester for 102 when it comes to the idea of having that primary signal and how that primary signal can elicit a ton of downstream events. Now we can think about it from the perspective of, this is going to be a negative outcome for the organism that receives whatever these peptides are going to be. And it can be a cocktail, meaning it can be a mixture of a bunch of different peptides because it messes up the signal transduction pathway that would be essential for different events that take place within the organism.
So when it comes to the nervous system, there are different anatomical divisions, and there is the central nervous system and the peripheral nervous system, which often is abbreviated as CNS or PNS. Now, the central nervous system contains the brain and the spinal cord, and these are both enclosed and protected by the cranium and the vertebral column, respectively. Now, notice that there's less going on downstream when it comes to the central nervous system beyond these two portions, and that the peripheral system has a lot more going on. So it's broken up into several divisions. So the peripheral system is essentially everything else aside from the brain and the spinal cord. So we're thinking about nerves and ganglions are just swellings of nerve tissue specific to the peripheral nervous system. And the peripheral nervous system gets divided into two divisions. So you have sensory and you have motor. When it comes to the sensory division, we are going to be carrying signals from different types of receptors, so sense organs and sensory nerve endings, and we're carrying that information to the central nervous system. So we're sensing something, and we're sending that information towards the central nervous system through that sensory division. Within the sensory division, we have our subdivisions. So we can have our visceral sensory division. So this would just be things such as viscera, thoracic and abdominal cavities. So things like the heart and lungs, which you would have discussed during your vertebrate dissection. And then we have the somatic sensory division. And that's going to be receptors in skin or muscle, bones, and joints. So in both scenarios, these are still divisions in which we are going to be carrying signals from receptors found in these locations towards the central nervous system for the central nervous system to essentially process and figure out what it's going to do with that information. When it comes to the motor division, that's going to be carrying signals towards muscle cells so that it can carry out the body responses. What should we do with this information? How should we respond? So there is the somatic motor division, and that will send signals to skeletal muscles. So things such as the muscles in your arms and legs, these will lead to muscle contractions, which we'll talk about in our discussion of motor mechanisms. And these are essentially things that you can control. So voluntary muscle. And you've probably had a discussion about the different types of muscle, cardiac, smooth, and skeletal in the histology portion of your lab. And then there's also the visceral motor division. And so this will carry signals to glands. It will carry it to cardiac muscle, to smooth muscle. And these would be examples of places that lack voluntary control. So with the visceral motor division, we can break it down even further into sympathetic and parasympathetic. Sympathetic, you're really thinking about fight or flight. And so this will lead to things such as accelerated heart rate, increased respiration. And if we're really focusing on, getting out of harm's way, it will decrease emphasis on things that are not essential in order to achieve that event. So something like digestion would be decreased. The parasympathetic division is a calming side. So there would be reduction of heartbeat, and then we could go back to providing emphasis to processes such as digestion. So within all this is just an overview of the two major divisions. You want to think about what's involved. Central nervous system, as long as you know that the brain and spinal cord are the things that are found there, then if we're talking about anything else, it's part of the peripheral nervous system. So know the different divisions, what we're utilizing, what we're communicating with in order to convey information. And so when it comes to the nervous system, it's really about sensing something, processing that information, and then commanding the body to lead towards some downstream response, whether we're focusing on muscle cells or gland cells. So we get the information, we process the information, and we do something with that information.
Concept 48.1, neuron structure and organization reflect function and information transfer. This is just an extended way of thinking about the relationship between form and function, and the neuron provides an example of that. Now, while this concept is compartmentalized within Module 1, it's definitely something that you want to continue to consider as we go through ideas like propagation of a signal, being able to transmit that information along the length of an axon. Why does the form of the neuron help to facilitate that process? How could manipulation of its form have an impact on its function? Where do we see changes with respect to the organization of the neuron and the role that it's meant to play? So that's really what the idea is behind concept 48.1. But honestly, I feel like this concept is one that really infiltrates the majority of this chapter and is one that you should apply throughout. So don't restrict your application of this to module 1. Think about how it extends all the way through. Here, I'd like to go over the basic properties of neurons. And so when it comes to neurons, it's all about receiving, conducting, and transmitting signals in that particular order. So it receives a signal, it conducts that information throughout its body, and then will transmit that signal, leading to some type of effect or outcome. So one major property is that of effective communication. Another property of neurons is that they are extremely excitable, which means that we can elicit a response, and we'll think about the different ways that neurons can be excited and what the downstream impact of that excitation will be. They also display a really significant amount of longevity, which essentially means that they can function optimally for a lifetime as long as there's adequate nutrition available. Neurons are also amitotic, so within that you hear the word mitosis. So essentially, upon achieving their role within communication for the nervous system, they lose their ability to divide. So amitotic, they're not going to go through mitotic division. So this means that neurons essentially can't be replaced if they're destroyed. And we find that these types of cells are located primarily in G0 of the cell cycle, so they're in a quiescent phase. That's not to say that all neurons lack the ability to divide. There are some neurons that can be replenished, but those will be ones in specific regions within the brain that will contain stem cells, and those stem cells are capable of maintaining a supply of neurons throughout their lifetime, right? So you've learned about the different properties of stem cells, so it would make sense that the presence of stem cells here, and these would be adult stem cells, are capable of giving rise to a continuous population of neurons, but not all neurons have a stem cell origin that's available. Another property that we find within neurons is that they have a really high metabolic rate. And because of this high metabolic rate, they need a lot of oxygen, a lot of glucose. We're thinking about, ATP production. So what that then means is that neurons would be highly sensitive to deprivation of those things. So just in the same way that our hearts are really sensitive to being deprived of oxygen for a few minutes, the same would be applicable to neurons because they are just constantly doing work in order to go through the process of communication. So anything that might perturb that would have a negative impact on the nervous system. So these are just kind of a list of properties that I'd like you to know as it relates to neurons as a whole. We're not really going to go into making really specific distinctions between different types of neurons outside of the basic categories. So this can be something that we can apply to the majority of the discussions that we'll have as we progress forward. Here we'll take a look at neuron structure and go over some of the basic vocabulary and the roles that they play with respect to communication. So the control center is essentially the cell body, which is also referred to as the soma. soma. And it's similar to other cells in the sense that it contains organelles. So there's a nucleus, which is indicated here, in addition to other organelles which aren't expressly shown but are present. And when it comes to those organelles, that can consist of mitochondria, lysosomes, Golgi. There's an extensive rough endoplasmic reticulum. This is a concept that is relevant to Bio 100 and thinking about if we have a ton of one type of organelle, what would that tell us about the role that's being played within a particular cell? So an extensive rough endoplasmic reticulum, we can think about a lot of protein synthesis there because of the presence of those ribosomes. But mature neurons will lack centrioles, and centrioles were a topic of discussion when it came to cell division. So mature neurons don't have the components that would be necessary for cell division to take place. Now, in addition to the cell body, there are processes or extensions called dendrites that we can see extending off of the body. And these short branching extensions, these are the input regions. They're going to provide a ton of surface area in order to help receive signals from other neurons, for example. And they can take those messages and convey them toward the soma. In addition, we can see right kind of at the end of the soma or cell body, there's an axon hillock. It's just a little bit broader than the axon. So the axon hillock will give rise to the axon or the nerve fiber. And when it comes to form and function, this is really specialized for fast conduction of nerve signals to points that are, distal or away from the soma. Within the axon, there's cytoplasm. It's sometimes referred to as axoplasm, which isn't indicated here. But then there is the membrane that's called the axolemma. So here it's saying axon, but there's axoplasm, which is the cytoplasm, and the membrane is the axolemma. And when it comes to the axon, we see that we're only showing essentially one, but some axons will branch extensively at their distal end. And when we take a look at the distal end here for the presynaptic cell, what we see is a bunch of branching, and there will be terminals that will end in a synaptic knob. So really just kind of a small bulbous region right at the end of that. And when we look at the bigger picture, it looks like the presynaptic and the postsynaptic cell are directly touching one another. But when we zoom in the illustration on the left-hand side, we see that there's a small space between the presynaptic cell and the postsynaptic cell, and that space is the synapse. So this would be the cell before the synapse, the cell after the synapse. Okay, so presynaptic and postsynaptic. But at the synapse, we can see that there's a small expanse of space in which these molecules that are referred to as neurotransmitters, they're going to migrate from the presynaptic cell to the postsynaptic cell. And these neurotransmitters can elicit some type of response. So that's kind of the basics of neuron structure with a little bit more vocabulary than what was shown here. But what you want to take from this is the ability for the presynaptic cell to communicate with the postsynaptic cell. It can do that through the release of neurotransmitters. But we also want to think about the effective communication and relay of a signal that occurs within a single cell, right? So how are we able to elicit that information along the length of a presynaptic cell? Or once the postsynaptic cell receives information on what it should do, how is it able to transmit that information along its length and elicit some response somewhere else? When it comes to this illustration, you can also see the unidirectional nature of the arrows, meaning that we're getting something from the dendrites, it heads towards the soma, goes along the length of the axon, reaches a postsynaptic cell, and we keep heading in this kind of forward direction. But there are things outside of the stimuli associated with presynaptic movement and the synapse release. And this is a concept that is sometimes lost in discussion of neurons because the emphasis lies so much on communication. But there are substances that can travel along the axon, and those substances can move in one direction or the other. So there can be anterograde movement, and that would be movement away from the cell body. So an example of that could be mitochondria, cytoskeletal elements, certain vesicles that are used to renew the plasma membrane, or enzymes that a neuron might need in order to generate neurotransmitters. And then there's also retrograde movement, and that will be moving towards the cell body. And that's normally organelles that are going to be returned in order to be broken down or recycled. So there are mechanisms of communication on an intracellular level, and that allows the cell body to know the status of the axon terminals. And some signals can activate nuclear genes in order to promote growth. So I don't want this conversation to be one that leaves you with the thought that we're only thinking about a neuron as a way of ions that are moving in one direction or another, eliciting some type of response. This is still a cell. And so the things that you've learned when it comes to the tour of the cell are still applicable here. And I just don't want you to lose sight of that as we continue our discussion, because our discussion will be primarily focusing on electrical and chemical stimulation. But these concepts are just as important too when thinking about neuron function.
So here is a slide depicting nerve tissue at 20x magnification. And when it comes to the neurons that we find there, there are 1 trillion neurons within the nervous system, and they make up a significant portion of nervous tissue. But even as we're looking at this image in which we can see our neurons here, the trillions of neurons that we would find within the nervous tissue are significantly outnumbered by cells that are playing a supportive role, which are called neuroglia. And those are these tiny punctate staining that we see throughout the slide prep. So we can see there's far more of these guys here than there are actual neurons that are visible within this particular section of slide. Now, when it comes to glial cells, they will be broken up into categories, but overall, we're really thinking about being able to provide support and protection for the neurons. We want to be able to hold neurons in place, and this ability to hold neurons in place is an important role that glial cells play because it can prevent neurons from interacting with each other in an inappropriate fashion, and it helps to ensure that the interaction that does take place is at the specific location where signal transmission should occur, and that allows for control of conduction pathways. It can also provide nourishment to the nerve cells and really just help to maintain that balance and homeostasis that's needed.
This image gives us an opportunity to take a look at different examples of neuroglia, because when it comes to the nerve cell tissue prep, what we find there is that it just looks like a bunch of dots, right? But in essence, glial cells actually resemble neurons in the sense that they have extensions, but they can't transmit nerve impulses, and they don't lose their ability to divide, so they're not amitotic. And a lot of brain tumors are gliomas. So thinking about that ability to replicate and divide and errors, things that are related to cancer discussions that you would have had whether you were in the morning class or within the evening class. Now, when it comes to the neuroglia, there are 6 types, and they have unique functions. There are 4 that are found within the central nervous system, and then we'll go through the 2 that are found within the peripheral nervous system in the next slide. So we're just going to go through each one of these one by one just to get a sense of the supportive role that they are playing as it pertains to neuron behavior and the nervous system overall.
Astrocytes are the most abundant cells in the central nervous system, and there's a long list here which is indicative of their diverse roles. Astrocytes have a star-like shape, so there's a bunch of radiating processes that will extend from the body. And astrocytes will have something called perivascular feet, and they will contact the blood capillaries and can help stimulate them to form. They can also form a tight protective seal, and that is referred to as the blood-brain barrier. So we've discussed blood in the sense that it has all these different goodies in there, and how these components help the body to do things like form clots or fight off pathogens. But we've also discussed that blood can be the site of bacterial toxins and other potentially harmful agents. And because brain tissue is pretty much irreplaceable, it's really important that it be well protected. So a blood-brain barrier strictly regulates what can get from the bloodstream into the tissue fluid of the brain. So the blood-brain barrier ensures that anything that's leaving the blood has to pass through the cells and not between them. And by passing through the cells, you can have a tighter level of regulation. So there's really good permeability of things like water and glucose, things that are lipid soluble, so gases like oxygen, but there is also a high level of permeability for things like alcohol and caffeine and nicotine. So that's something to keep in mind when it comes to those types of substances as well. Now, when it comes to the blood-brain barrier, another thing that's relevant from earlier discussions is that it is really not an effective means of medication delivery for a lot of chemotherapies. So a lot of chemotherapy or cancer-based drugs can't pass through the blood-brain barrier. So that makes treatment options for brain cancer really complicated. So other things that the astrocytes can do is monitor neuron activity, and that can lead to stimulation of dilation and constriction of blood vessels, which allows for blood regulation in brain tissue due to changing needs for oxygen and nutrients. It can help to regulate the composition of tissue fluids, and it can do this by taking up potassium ions and taking up neurotransmitters that have been released by neurons when those neurons are transmitting signals. And this helps to prevent those items from reaching really high levels. Another thing that you can see within the bottom of this list is the idea of injury. So when neurons are damaged within the central nervous system, astrocytes will form that scar tissue and fill that space that had contained those neurons. So nerve regeneration or neuron regeneration within the central nervous system is not something that is really, effective due to the scarring response that occurs within this area. Next are ependymal cells. These are ciliated cells, and they'll have various shapes, but they will be found lining the central cavities of the brain and the spinal cord, where they form a permeable barrier between the cerebrospinal fluid that fills those spaces. And the cerebrospinal fluid, or CSF, is a liquid, and that bathes the central nervous system. It fills those internal cavities, and the cilium movement of ependymal cells helps to circulate the CSF. And the CSF helps to cushion the brain, cushion the spinal cord. It can provide buoyancy to the brain, help protect it from injury. It can provide chemical stability, and that's important because deviations in certain components can disrupt activities that can lead to loss of control of body temperature, blood pressure. So cerebrospinal fluid is really important. And so these will be the supporting cells that are found within that region. Next are oligodendrocytes. And oligodendrocytes will insulate nerve fibers with a covering called a myelinate sheath. So here it says myelinate central nervous system axons. And myelination is important with respect to propagating signals along a nerve fiber. And the composition and specific role that myelin sheaths play within the axon will be discussed in a separate image. And the oligodendrocytes also will provide structural framework. So we're still supporting this overall idea of glial cells being supportive, and essentially glial, you're thinking glue.
Last, we have microglia, and microglia are small macrophages, and they develop from monocytes, which is a concept that was discussed in relation to hematopoiesis. Now, these particular types of cells will wander through the central nervous system, and they'll put out projections in order to sense for things like cellular debris or microorganisms or anything that really just shouldn't be there. And they'll go through the process of phagocytosis, which is a concept that you learn when discussing the immune system, and that will help to ingest and hydrolyze whatever they are picking up. Now, as one would expect, we would see concentrations of these particular types of cells in areas that are damaged by things like infection or trauma. So we can actually get a sense of where injury is found based on varying concentrations of microglia. So if there are clusters within the brain tissue, that gives us a sense of where the damage is located. And one last thing is that when it comes to microglia, their presence here is important because the cells of the immune system that you've previously learned have really limited access to the central nervous system. So there's a reliance upon these types of supportive cells in order to achieve these events. Within the peripheral nervous system, there are satellite cells and there are Schwanns.